All right, let's jump right into this explainer. Today we're diving into the dramatic, high-stakes lore of cell division to see exactly how one basic, everyday cell gets a massive glow-up. We're talking about a transformation so epic, it literally creates the foundation for all biological diversity. So grab your notes, because we are unpacking the ultimate cellular remix.
Seriously, if you've ever looked in the mirror and wondered why you have total main character energy instead of just being an exact, copy-paste NPC clone of your parents, the answer is a cellular process called meiosis. It is the biological reason you are completely unique, built different, and definitely one of a kind.
So what is this process exactly? Think of meiosis as the ultimate character arc. According to the source material, meiosis is a process specifically used for gamete production. Those are your reproductive cells, and spore formation. We start with one standard diploid cell, which scientists call 2N, because it has two full sets of chromosomes. Then, it goes through two intense stages of division to produce four totally unique haploid cells, or N cells, with just one set of chromosomes. It's basically taking your genetic inventory and having it, but in the coolest way possible.
Stage 1. Meiosis 1. The genetic remix. But before the cell can even think about dividing, we need to introduce the stars of the show, which are the homologous chromosomes. In a diploid cell, these are pairs of chromosomes that are totally matching fits. They're similar in shape, and they carry the exact same types of genes, with one inherited from each parent. They give off matching vibes, for sure, but if you look closely, the specific details, the alleles, are actually slightly different.
Now, the defining event here is called crossing over. During this first stage, these homologous chromosomes get super close and literally swap pieces of their DNA. This creates genetic recombination. It's exactly like two artists dropping a massive collab track. They trade verses, mix their styles, and what you get is a brand new, genetically unique sequence that has literally never existed before in the history of the universe. It is biology's greatest remix.
Watch this drama unfold from prophase 1 to telophase 1. First, in prophase 1, those homologous chromosomes pair up and do their crossing over collab. Then, in metaphase 1 and anaphase 1, those newly remixed chromosomes are divided into two distinct groups and pulled to opposite sides of the cell. Finally, we hit telophase 1, where the cell actually splits, leaving us with two haploid cells. The chromosome inventory has officially been halved.
Okay. Here is the absolutely crucial point, and I need you to lock in right now, because biology teachers love to gatekeep your A+ with this exact trick question. In meiosis 1, the homologous chromosomes are what divide. The sister chromatids, the two identical halves making up that X shape, do not split up yet. They stay firmly attached. Do not get this twisted on your exams.
Stage 2, meiosis 2, sister chromatids split.
And here is the best news you're going to hear all day. This source material drops a massive hint here. Meiosis 2 is exactly like mitosis. Literally. If you've already studied standard cell division, you already know the vibe of how this second stage works. The assignment is understood. It's basically copying the homework but changing it just enough so it looks different.
So, those two haploid daughter cells from the first stage are now going to undergo another division, but this time, it's the recombined sister chromatids taking center stage. Remember, these aren't your standard, boring sister chromatids anymore. Because of the crossing over in stage 1, they have been uniquely remixed.
Moving through prophase 2 and metaphase 2, everything lines up. Then, the main event, anaphase 2 hits, and those recombined sister chromatids finally separate, getting pulled to opposite poles. We finish up with telophase 2, completing the split and resulting in the creation of four final haploid daughter cells. The solo era has officially begun.
All right, corporate needs you to find the difference between these two stages. Pay close attention here. In meiosis 1, it's the homologous chromosomes that divide, ending with two haploid cells. But in meiosis 2, it's those sister chromatids that finally separate, ending with four haploid cells. If you can spot this difference, you have officially mastered the core logic.
The haploid glow-up finale.
The magic number at the end of this epic cellular journey is 4. Exactly 4 haploid daughter cells are produced from that single original diploid parent cell we started with.
But they aren't just any normal cells. Our source material emphasizes a massive point here. Because of that crossing over and genetic recombination we saw in the first stage, all 4 of these daughter cells are completely 100% genetically unique. They're different from the parent cell, and they're genetically different from each other.
Every single one is a literal one-of-one rare drop, no cap.
To wrap up this explainer, I want you to think about this. If meiosis guarantees that every single one of these reproductive cells is a completely custom remix, how does that infinite variety shape the future of all living things? It's the reason life adapts, evolves, and survives. So the next time you realize you're built different, you can thank meiosis for giving you that main character energy.